in healthcare. Okay, so this is our company is located. So our company is mainly deals with genomics, proteomics, and metabolomics, all kind of omics technology. So that is the reason in my company name itself I have mentioned Insilicomics. So Insilico represent as a dry lab, omics represent as genomics, proteomics, and metabolomics, on all kind of omics technology we do in our lab. So, which is located in Ooty. It's a main place in Ooty itself. It is very near to Charing Cross and near Rose Garden, our office. Because basically, I'm from Ooty. That is the reason I have started my company in Ooty. And we do a lot of research on different fields of bioinformatics. And coming to the next slide, mainly we deals with drug designing in our company, project related to docking and dynamics, and we do NGS data analysis related to omics data analysis of genomics, proteomics, metabolomics, and all kind of different ideas. And we do programming mainly on Python and R programming. That's what we do in our company. And when it comes to the milestone, actually we registered a company in 2021 as a name called Omics. So in 2021 we started. In 2022 we renamed as Insilicomics. And 2023 we have completed more than 450 projects, and as well as 100 students for internship. We provide internship and as well as different areas in 2023. And successfully we signed 10 MOUs with universities, so in 2024 as of now. So when it comes to the achievement, so every year we get a lot of students for internship, not only internship, even projects, because I have shown here only for internship. So as of now we have completed 224 students for internship and training, so on students project. And so these are our numbers where and all students have come and all these things. We get students from Tamil Nadu, Bangalore, Karnataka, and Hyderabad and Kerala, different places. We do get students for internship. And when it comes to our major topic, so that is my introduction about our company. I'm not going to talk about much. I'll talk about what is omics data, and what is NGS sequencing, and project related to omics data. That is what today is my major topic. So yeah, what is omics data analysis? So the thing is, I hope most of the people are not may are aware of data analysis. So if we consider any data, we need to do the data analysis. Okay, so when it comes to the omics data analysis, so majorly what is omics data in the sense, for example, we have omics data. Okay, that omics data represent, it may be a genomic data, or it may be a proteomic data, or it may be a metabolomic data, or secretomic data, whatever. If it is in genomic data, means we can say it is a gene. Okay, it may be a nucleotide sequence of maybe DNA sequence or RNA sequence. In RNA, it may be a siRNA, miRNA, mRNA, tRNA, rRNA. All these data, it is called genomics data. So that data can be stored in computer. We have to do the statistical analysis. So the major thing is, if we consider here genomics data, computer science and statistics is called genomics data analysis. When it comes to protein data, it is called proteomics data. It may be a mass spec data, or it may be a like structural data of protein sequences. All these things comes under proteomics data. Then it is do computer science for storing the data and analyzing the data and using statistical method. So this combination is called proteomics data analysis. And when it comes to metabolites, for example, we get a lot of metabolites from the plants or human gut microbiome. There are different places. So all these metabolites data can be stored in computer, can be do the analysis using statistical model. That called metabolomics data analysis. When it comes to all these things, we used to call as a multi-omics. That's what they used to say, multi-omics. Multi-omics means proteomics data or metabolomics data or secretomics data or genomic data, whatever it is. All these things can be stored in computer, can do the analysis of statistical model. So these three combination called omics data analysis. Okay, so the omics data analysis, it's a recent trend. Previously, they used to call bioinformatics. So the point is, here we have a molecular biological data can be stored in computer, can do the analysis of statistical model. That is called bioinformatics. The new version of bioinformatics is nothing but omics data analysis. So that is what we do in our company. We do all kind of omics. In computer we'll do the statistical analysis. That is what the major concept of omics data analysis, because the omics data is boom now. I can give you one very simple example. In this image they have shown one laptop. From the laptop there is a one dna comes, there is a one cloud. In that cloud there is some servers are there. One dna goes inside, the dna sequence comes outside. Okay, actually speaking, this particular image I have taken in 2000 maybe 14 or 15 time. 2014-15, they may not aware of much about what is cloud computing and what is sequencing much, all these things. But this image I have taken in 2012 or 13 time. The point is, on that particular time itself they talk about cloud computing. So nowadays, using your laptop you can able to connect cloud, example Amazon cloud or Google cloud, and you can do the sequencing data analysis using cloud computing. So the advanced techniques are happening nowadays. Using your laptop it's enough. You can connect your servers and you can perform everything. We have done lots of project on omics data analysis. That is what I want to show here. When it comes to the major step of earth biogenome project, okay, I just want to interact with the participant. Can you tell me what and all the organisms are present here? Can anyone uh, can anyone tell me what and all the organisms are present here?
Plants, snake,
One
animals.
second, sorry. Yeah, one second, I'll switch off my video, otherwise it will become very slow. Sorry, I'll switch off my video. Yeah, okay. Son, fine. So, anyone, can you tell me what and all the organisms are present here?
Plant, bird,
Okay,
snake, deer.
perfect. Yeah, perfect. So now I'll give you some idea. Please select any one of the organism, and we can discuss a little more about the organism.
Plant,
Yeah, which plant? Can I say it's a grapes? It looks like grapes plant, right? Just consider. We'll talk about the grapes. Okay, so the major aim of this project is Earth Biogenome Project, in the sense, whatever the organisms are present in that particular Earth, they'll do genome sequencing. Example, consider this particular grapes. It is a dark violet color grapes. Okay, and this particular grapes located in Karnataka. Just consider this particular grape is located in Karnataka. Okay, I want to check the similar kind of grape may presence in Tamil Nadu also. Similar kind of grape may presence in Rajasthan also, or similar kind of grape may presence in Africa also. Am I right? It is common. Correct. So now what I am going to do now, I'll take the entire genome sequencing of this particular grape. It may be a whole genome sequence, or it may be a transcriptomic sequence or metagenomic sequence, whatever the sequence. I may have the whole genome sequence of Karnataka variety of one particular grape. Okay, the same kind of plant may presence in Tamil Nadu also. I'll take a grape, I'll extract the DNA, I'll do whole genome sequencing. Then I can take from Rajasthan, I'll extract the DNA, I'll do sequencing. In Africa, I'll extract the DNA, I'll do sequencing. Now we have four grapes genome sequence. Okay, now what I can do, I can do the comparative genomics. Why I have to do comparative genomics? So we can able to find out what conditions are responsible for growing this particular plant. So this is everybody they do. Any NGS projects, any genome sequencing project, they'll do this. Then what new? The thing is, Earth Biogenome Project means everybody they'll do one organism. But nowadays, their planning is the entire biogenome project idea is whatever organism is present, they want to do genome sequencing. So when the genome sequence improved lot, automatically huge number of data will generate. When the huge number of data generated, we need to store somewhere, someone need to analyze. So the analysis persons are bioinformatician. So that is why most of the industry, they started with bioinformatics. Okay, I just talk about just only one particular plant. If you take snake, if you take panda, if you take anything, fish, whatever it is, everywhere they'll do genome sequencing. And one more important point, in Karnataka itself, in Mysore, similar kind of plant may present, in Sirsi, similar kind of plant may present, inside the states itself, they'll do sequencing. So it will make huge number of data. The data is very, very important to the coming to the next process. What I'm talking about here, it is called biogenome project. Please try to understand. I want to explain one important thing. In the omics technology, genomics already came to the market. Why it came to the market? Because of human genome project. I'm going to explain later. It's already there in the market. So proteomics and metabolomics, it is starting. Proteomics in the research field, metabolomics is recently they started. Okay, so most of the industries, they started with genomics. So that is the reason the genome project have started. Definitely, Earth proteome project may start. The Earth metabolomic project may start. So that has become Earth multi-omics project. It can start in soon. So that is the things I want to explain. What are the importance of genomics? And coming to the next slide, this is one of the example image what I have shown here. Earth biogenome project, which whatever organisms are identified, they'll do sequencing. Okay, it's like one organism is one PhD scholars project. So for example, if you consider snake, it's a one PhD scholar project of that particular area. Maybe in Karnataka, in maybe in Mysore. In the same kind of snake, maybe doing sequencing in Sirsi area. So they'll do one more sequencing. And in Tumkur area, maybe in Bangalore areas. Like one projects can be done, but entire thing has become a big bio project. It's a biogenome project. So I hope you are clear with this concept. So till now, if you have any questions, please ask, because after that I'll go with the NGS sequencing techniques. So please, you can interact with me. Yes, any other questions?
Sir, when we are analyzing the sequences of a species found in different regions, do you
Yes.
find any difference?
Yes, we find mutations. We found just which genes are highly expressed, which genes are low expressed, and what condition that particular genes are expressed. That we do in a transcriptomic projects. So RNA sequencing also we do, DNA sequencing also we do, even epigenetic changes also. Yes. Yeah. Any other questions?
No, sir.
Yeah. Fine. So then we'll start with the next process. It is called NGS. It is very old. I'm talking about NGS. Most of the people knows about what is NGS. Even though I'll try to explain one concept. Okay. So, as you know, sequencing of human genome project. I hope maybe most of the I guess all the participants are faculties. Am I right? So even I was a faculty. Almost I worked for almost 12 years as assistant professor, and every genetic subject or any subject, we used to ask one question. 5 mark question is nothing but human genome project. Student, they'll write basic information about human genome project started in the year 1990 and completed in 2013 and 2001, like that they'll write. But okay, but what is the application? That's most important, right? See, I'm going to give one example with human genome project. Actually speaking, in 2001, okay, so in 2001, there is a human genome project was completed. Actually, the draft genome was submitted. They spent approximately $2.7 billion. Okay, that means they spend approximately $2.7 billion. So participant, anyone, can you tell me how much amount in Indian rupee? Can you calculate and tell me? $2.7 billion. Anyone, please. Now the dollar rate is 80 rupees, but just consider in 2001 the dollar rate is 50 rupees. I want to know. I want to be in a crores. How many crores?
So 2 crores something. 2 crores 14 lakhs
Oh,
something.
only 2 crores. 2 crores is
Oh,
nothing.
no, no, no. 200 crores, I
No,
think.
actually, it's a 2,000 crores. 2,700 crores they have spent.
Okay.
Approximately 2,500 crores. Please try to understand. The government of US and UK and Japan, they have spent approximately $2.7 billion, in the sense approximately 2,000 crores they have spent for one project. Okay, and it took approximately 11 years. It's very important. It took 11 years. Okay, and this project was done by government project. It's actually human genome project. This project is a government project, which led by, there is a professor named called Eric Lander, so who's a director of Broad Institute, which is located in Harvard University, MIT. So the thing is, this particular project was published in Nature Journal with a cover page in 2001. So the same time, there is a one more company name called Celera. Okay, there's a company called Celera. They have done the human genome project in just 3 years. Just 3 years they have done the human genome project, and they spent only $100 million. And they have completed in 2001 itself, which came in Science magazine cover page. Okay, so what happens, actually there is a one scientist, he left human genome project, he started his own company, and he can able to complete within the 3 years the same human genome project. Okay, and which was published in Science magazine in a cover page. So when the 2001 comes, so everybody they talk about biotechnology and future of biotechnology, because it came in Nature cover page. Everybody they talk about human genome project is completed, what to do, and all these things they talk about. Okay, so in 2001 they have talked about one word called personalized medicine. I hope all of you heard about personalized medicine. Yeah, so they talk about personalized medicine, but the problem is, in 2001, if I want to do my genome sequence, for example, if I'm some person has cancer, the doctor is prescribing the technique. For example, you do genome sequencing and come, and will identify the mutation and which drug suits for your body. That is what the idea. Okay, if the person goes to any research centers like Celera or Human Genome Project Research Center, if they provide the sample, they have to wait for 3 years, or they have to wait for 11 years, and they have to spend billions of dollars. Within a 3 years, the person may die, right? Then what is the use of the technology? The idea was good, but at that time, the technologies were not up to the mark. So to coming to the overcome that particular project, the problem, they have come up with the one concept name called NGS, next generation sequencing. Why these two techniques were very slow? Because they have used Sanger sequencing technology, which is chain termination method, and which is very slow, but it's more accurate but very slow. Okay, coming to the first next generation sequencing instrument name called 454. Okay, which is later, there's a company called Roche, they have bought this company called 454 in 2001. They have done the human genome project. Within a 3 months they can able to get the entire genome, and they have to spend $1 million. Within a $1 million you can get your entire genome. So compared to human genome project, private and what does it means, private and government, this particular project is very faster, and you can get your genome little faster manner. So when it is in 2007, using these techniques, we can able to get the project, means the genome sequencing is lesser price, lesser month. But $1 million is not lesser price for normal people. If you are a billionaire, $1 million is cheap. If you are a millionaire, $1 million is very costly. Then we have to reduce the amount, and we have to reduce the month. Again, one more company name called ABI Solid. They have come up with the concept called, within 2 weeks you can get your entire genome and $60,000. Again, $60,000 is not for normal people. Am I right? For not for middle class people, for higher people. So again, we have to reduce the amount. So there is a one instrument name called Illumina, which is very very important instrument nowadays. In 2009, they have talked about $40 to $50,000. Within a 1 week, you can get your entire genome. Okay. In 2016, there is a one instrument name called nanopore sequencer. So this is actually, it is a very very smaller genome sequencer, and it is a real-time genome sequencer. Okay. So just $1,000, you can get your entire genome within a 1 day. So look at the drastic changes. In 2001, this was approximately, we have to wait for 11 years to get the entire genome. But now I'm talking about 2016. Within a 1 day, you can get your entire genome and $1,000. Okay. So in this slide, if anybody is having question, please ask. Because based on this slide, I'm going to explain some of the important points. So anybody is having questions?
Sir, the last
Yeah.
research that is in 2016, as you said, through this
Yes.
Oxford Nanopore,
Yeah.
they are doing it at a very cheaper cost, and even the time consumed is very less.
Yes.
Is it reliable when compared to the
Yeah.
earlier ones?
Yeah, your question is exactly correct. I will tell you now. So as of now, the people are telling Illumina is very good. I will tell you why. Illumina sequencing is very good compared to Nanopore. Am I right? That is what generally people they'll say. Illumina is good compared to Nanopore. Now I'm going to compare with Illumina versus Sanger. Which will be good? Anyone can you answer? Illumina is good or Sanger is good?
Sanger is good, sir.
Yeah,
Sanger.
Sanger is good, right? Because the accuracy level is 99.9% accuracy. Okay, Sanger. But it is taking lot of time. When it comes to Illumina, 99% accuracy. Okay, when it comes to Nanopore, it is 98% accuracy. Okay, so whenever you do the comparison, if you compare with Sanger, definitely Sanger is best. But we cannot rely on Sanger now because it is taking lot of time. See, if any person goes to doctor, they will say do sequencing and come. If you go for Sanger sequencing, again I have to wait for 11 years. Who will wait? Then wherever the technologies were little good and the sequencing little faster, so they'll go with that. So they'll go with Illumina. Yes, Illumina is good compared to Nanopore. Okay, now Nanopore is good, maybe compared to another instrument. Example, I can say PacBios. That is another instrument. And one more thing is Nanopore is a real-time sequencing. It is called SMRT sequencing technique, smart sequencing technology, single molecular real-time sequencing technology. Once you insert your sample in Nanopore, directly you can see your genome sequence. Okay, that much faster. But only thing is they won't do much amplification. The error rate is little more compared to other instruments. I hope you're clear.
Yes,
I
sir.
hope I have answered your question.
Yes, sir. Yes, sir. Thank you,
Yeah,
sir.
thank you. Any other questions like this? Okay. So I'm going to give one small task for you people. I hope all of you have a mobile. Please open mobile and then check. There is a website called 23andme.com. Please check. There is a website called 23andme.com. 23 ampersand and, that means and, ampersand and me.com. Have you opened the page? 23andme.com. Yeah. Can you check? Is there any offer prices there?
But it shows error, sir.
Oh, it's showing error.
Yeah.
1 second. I'll check once.
Yeah.
Every
Yeah, $99.
Okay, yeah. Someone is getting $99. $99 of what? Human genome, right? Genome sequencing. Am I right? Just $99, you can get your entire genome. And what and all the ancestral information, what type of disease you may get, all this kind of informations can be given in just $99. That means $100. Okay, approximately how much? Can I say 8,000 rupees in Indian money?
Yeah.
8,000 rupees, you can get your entire genome. and within a 1 day. That means that much drastic changes happens in the genomics field. And 23andme.com is one of the company. They do genome sequencing for the whatever the samples they provide, and they'll do sequencing a little faster, and they'll provide that what type of disease you are going to get, like precision medicine kind of concepts. Okay, so which is located in US. um, Recently means located in US, and then they have done the extremely good work on genome sequencing. But this particular genome sequencing, it's not for Indians, because this company is mainly located in US. It is mainly US people, okay, and Europe people, not for Indians, because India has one particular thing called genomic law. So our genome sequence should not go to other countries. So that is the reason we have a genomic law, so the data won't go there. But in 23andme.com, there are some Indian samples are there. That is nothing but those who are staying in US, they can do genome sequencing. Okay, there is no problem. Then what about Indians? If I want to do my genome sequence, what should I do? I want to explain one important thing. 3 months back, I guess 4 months back, there is a one company called Strand Life Science. I hope all of you heard about Strand Life Science, which is located in Bangalore, one of the biggest genomics company or NGS company. The Strand Life Science was bought by Ambani. So Ambani bought Strand Life Science, and they are going to make just 12,000 rupees for genomic kit. Within a 12,000 rupees, you can get your entire genome. So they have started in Mayancha Tech Park in Bangalore. 1 month back, they have got the inauguration. Okay, some of my colleagues are working in same Strand Life Science. It's a very big company now because they do a lot of research on genomics. Okay, like that, if you see in India, there are more than 1,000 companies are there related to genomics. Okay, so that's why the genomics is boom now. And one of the companies Insilicomics also. So whatever we do, genomics. Okay, so that is what happens now in the field of genomics. I hope all of you clear with the slides, right?
Sir.
Yeah. So the applications of
Sir,
NGS. Yeah. Yes.
Strand lifestyle is only for cancer detection.
No, 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 no. Now they started with everything. Common thing, because now
Because
we,
when I'm searching in net, it's showing only for like oncogenic treatment selection and all.
no, no, no. Yeah, it's actually as of now 12,000 rupees. They are planning to start in
Okay.
future, because recently Ambani bought Strand Life Science. So their idea is like they want to make a like 23andme.com website is there now. Similar way they want to make for Indian samples, and as well as they Strand Life Science, they deal with cancer, and they deals with diabetes and different areas. I can say
Even for COVID-19 also.
yes, COVID-19 also, they have done the genome sequencing. There is another company called Genotypic, which is located in Bangalore. They also do a lot of genomics. And they started with microarray, then they moved to genomics. And there is a company called Kaivor Genomics located in Chennai. They do cancer genomics. So I worked in the company. I hope when ma'am has introduced me, I worked as a senior bioinformatics analyst in the company called ADD License Private Limited. That company renamed to Kaivo Genomics, which is located in Chennai. So they do mainly on cancer genomics. Okay, so they do whole genomes, whole exome sequencing data for the tumor samples. Okay, they'll identify the mutations. They'll prescribe that means they'll give the reports to the doctors. Doctor will prescribe the cancer therapies. So almost that company is more than 6 7 years old company. Okay, so like that, Strand Life Science also they do. There is a one more company called Foundation Medicine. So I hope all of you know about Bill Gates, right? So Bill Gates invested in Foundation Medicine company, which is located in US. Okay, so the genomics is boom now. Everybody they talk about genomics. Okay, so even I want to explain one important thing. As a industry person, I have conducted many places the genomics workshops. Recently, I had been to Mahatma Gandhi Medical College in Pondicherry. medical college, I'm talking about medical college, I have conducted clinical genomics workshop for doctors. Please try to understand. I have conducted clinical genomics workshop for doctors. For what? Because most of the hospital they started purchasing genome sequencing. Okay, recently, 4 months back, Apollo they agreed to do genome sequencing. Apollo hospitals, they made it mandatory for genome sequencing. Okay, every hospital they have genome sequence, they have a clinical sample, they have a genomic sequencer, they'll do genome sequencing, but unfortunately they don't know how to analyze. So now the doctors are learning how to analyze genomic data. I hope all of you clear with this concept, what I'm trying to explain. The doctors are learning now what genomic samples, because they have a clinical samples, they have ethical approval, they'll do sequencing, they don't have any idea about analyzing the data. So for that, I have conducted a lot of training program on Pondicherry Medical College, and as well as Ramachandra Medical College in in Chennai. I have conducted many workshop on clinical genomics. Okay, related to different fields, and even I have conducted in Osmania University, which was in Hyderabad. So there also we have done a lot of training programs. Okay, so this is what majorly on cancer genome. Human Genome Project completed in 1990, and the publication of first NGS method 2005 is nothing but Roche 454, and there are many projects related to cancer. It started. Okay, so I hope all of you know about this. So the 1 second, yeah, this particular slide is most important slide. We have a sample, we have a sample preparation, we'll do DNA extraction, sequencing, reference, pipeline, everything is done. But only the important thing is clinical interpretation. So we get a lot of data, but how to interpret it in the clinical way? So that is very important step in NGS data analysis. Okay. See, this everything can be done in AI nowadays. AI can do, sample preparation cannot do, but some instruments can do automatically. DNA extraction also automatically it can do. Sequencing can be done. Pipeline, all these things can be done in AI, but clinical interpretation AI cannot do. Manually, we have to do. For that, you should have a basic clinical knowledge. Okay, that's most important. It generate a lot of data. We need to do the interpretation. That is what I want to explain, one important thing. And when it comes to this image is very important image. This image is based on proteomics. Okay, the world's first proteomics human proteome was done by Indian scientist. Okay, the scientists from Institute of Bioinformatics. I hope all of you know about IOB, which is located in Bangalore. They have published human proteome map in 2014, which came in Nature cover page. The same image, it came in Nature cover page. Okay, what they have done, they have extracted each and every tissues. They have extracted the protein. After extracting the protein, they have extracted, they did XDS phage, trypsin digestion, HPLC, and mass spec and data analysis. They have identified how many proteins are present in human genome. The human, not genome, human genome project is how many genes are present. Human proteome, how many proteins are present. And next one is metabolome, how many metabolites are present. If you take all these three, it has become human multi-omics data. I hope all of you clear with this. Yeah. Any doubt in this slide? Anyone, please, if you have any questions. So how much time I have, ma'am? Hello?
So till
So the
one.
session is up to 1 o'clock.
Okay,
Till
fine.
1
Thank you.
o'clock.
Thank you. Yeah. Then I'll just start with basics about genome sequencing. So this is again proteomics image only I have shown here. And when it comes to NGS platforms, Illumina, iSeq, MySeq, and Iron Torrent, Iron Proton, there are different instruments are available. But nowadays, Iron Torrent, and GS Fluxes, all these instruments are outdated. Only people are using Illumina and Nanopore and PacBIOS. So these instruments are commonly used, but in India, most of the places they have Illumina sequencer. That is very common nowadays. The accuracy is very high. And when it comes to the proteomics platforms, so the instruments like Arbitrop, Qtrop, and there are instruments like mass spec, LCMS, MS, and there are different kind of ion exchange chromatograph. There are different type of mass spec data as a sequence are available. The instruments are available. And when it comes to genome sequencing, so generally what we do, we'll take a genome and we'll do the next process called fragmentation. We'll get a lot of short fragment of DNA. That short fragment of DNA goes for sequencing. So generally, the sequencing is nothing but, I hope all of you know about the sequencing. It is Sanger sequencing, but we can use recent sequencing technology, and it is generated the sequence, short DNA sequences. That short DNA sequence can be analyzed using computer, so that computer data produce assembly and final. I hope most of the people they deals with the final sequence. They'll go to NCBA website, I'll download the sequence, and they'll do the analysis. But as a bioinformatician, what we do, we'll take a short DNA sequence, we'll do the assembly, and we'll do the annotation, and finally the final sequence will come. So for that, I'm going to give some example projects. So this project we have done uh, on oral cancer sample. Yeah, am I audible now? Yes.
Yes.
Yes, ma'am.
Yes. So this
Yes,
project
ma'am.
mainly based on oral cancer data. So this is we have done when I was in a college. I gave this projects to my students. So they have done this project. I'm just explaining the methodology later. If time permits, I'll explain the results also. So we have taken the oral cancer exome data for the 12 samples, which is published in PRJBEV website. That means Bioproject ID. Then we have extracted using some softwares, and we used trimming. So if the raw data is not good, we have to go for trimming. Then we have done the quality checking, and we have aligned with the reference genome of human genome. So human genome 38th version we downloaded, and then we have aligned our raw data to the reference genome using, there's a tool called BWA, and we identify the mutations using GATK pipeline, and we identified SNPs, and we annotated SNPs using ensemble variant predictor, and we have done the molecular dynamics simulations concepts. Okay, so one of the project what we have done, so I'm not sure that image is there or not. I'll just check once. It's become slow. Yeah. Okay. This is a one methodology, and one more project we have done. I'll show this. I hope maybe all of you know about that. If anybody goes to forest or anywhere, they will see some plant. Am I right? So once they see the plant, they'll get some question: is this plant is a new or not? That is a one question they used to get. Yes, it is a new plant. As a biotechnologist, what you will do? Can anyone say? You went for trucking to the forest. You found there is a one particular plant is very new. It looks like very new. Then what you will do as a biotechnologist? I know. Yes, please.
Genome sequencing.
Oh, directly
They
genome sequence
analyze with gas chromatography.
inside the
Okay.
forest itself.
So we'll
Okay.
collect the samples. We'll
Oh,
collect the
perfect.
samples.
So that means we don't have that much facility. Am I right? So first thing is what we have to do? We have to collect the samples. Am I right? Once you collect the samples maybe you're going to collect the sample maybe root or leaf or entire plant. It depends upon you. Okay, I collected the sample. I came to my lab. Again, I have one more question. What kind of analysis I want to do? Okay, now I have one small question. Most of the people, what they'll do, if it is a medicinal plant, what they'll do? What kind of analysis they'll do if it is a medicinal plant?
Try to
Trying
extract
to
the
analyze the components of
phytochemicals.
particular plants.
Yes, perfect. So that means it is nothing but phytochemical studies. Am I right?
Going
Correct.
for gas chromatography.
First, they
Yes,
go
yes.
for phytochemical studies.
Yes, yes, gas chromatography and like Sox slit apparatus to extract that phytochemicals and all these things will do. But actually, we hate that particular topic. Why? I will tell you. As a bioinformatician, generally everybody they'll do because I'll explain this because we have done this project in 2014. Okay, uh, this 2014 we have done this project. Every students they'll come up with the concept. Sir, I want to do phytochemical studies. I want to do phytochemical studies. I gave that idea. Don't go with phytochemical studies. Go with some other technique which is new. And that time the new techniques were transcriptomic. So in the transcriptomic, what we have done, we extracted the DNA of that particular plant. Okay. Once we extracted the DNA or RNA, we have to go for sequencing. Now tell me which sequencing we have to go. Sanger or recent sequencing? Anyone?
Sanger sequence.
So Sanger sequencing means I have to wait for 11 years. Is it okay? Yeah, your point is correct. Sanger sequencing is good, but I have to wait for 11 years. Because human genome is like 11 years. Then I have to go for NGS. Am I right?
NGS. Okay, sir.
Yes. In NGS, I can go Illumina sequencer. Yes, perfect. So we can go for Illumina sequencer. That sequence will generate, there is a one particular name called raw data. Okay. This raw data need to check lot of analysis. As a bioinformatician, what we'll do, we'll take a raw data and we'll do lot of analysis. First step is this raw data is good or bad? We need to check. To check that, we are going to use, there's a tool called fast QC. Okay, once the raw data is good, we have to go for de novo assembly. I'll tell you why we went with de novo assembly, because when we started this project, that particular genome was not available. The particular plant genome was not available. That's why we went with de novo assembly. So here there are some lot of circles are there, right? It looks very good, but we spend a lot of time on that. I'll explain later. So which Kmer value is good, that I'm going to explain later. Then next we have done the functional annotation for that particular project. After that we have done the pathway analysis. Okay, why we have done this? Look at this. In this entire workflow, the top option is nothing but wet lab, bottom option is nothing but dry lab. If without wet lab, we won't get any raw data and we cannot do bioinformatics. And without bioinformatics, you cannot conclude the project. You cannot conclude the project. Only what you can say, I have done the sequencing, that's it. Okay, so that means your dry lab and wet lab is both are important. Okay, okay, fine. So what is the idea of this particular project? Anyone, can you tell me what is this, these things? Anyone? Where I'm showing the mouse? Please reply. Can I say it's chemicals? Secondary metabolites? Okay. Yes, sir. Now my question is, secondary metabolites comes from where?
Plant.
So can I say primary metabolites? Okay. Primary metabolites comes from where? How it is producing?
From some metabolic pathway.
Yeah, metabolic pathway. In the metabolic pathway, which is very important to produce this kind of chemicals?
Substrate.
Can I say enzymes?
Yes.
Substrate along
Yes.
with enzymes.
Enzymes. Okay. Enzymes comes from where?
From mRNA.
mRNA. mRNA comes from where?
DNA.
Okay, so what we have a DNA or RNA. So we can able to find out what and all the genes are responsible to produce these chemical molecules. Okay, that is what our project. So we have not gone with take this chemical and work. So our idea is what and all the genes are responsible to produce these chemicals. Please try to understand this project we have done in 2014. So in 2016, I had been to Dubai conference. I presented this work. So they were appreciated with the work, but they have said how you're going to conclude exactly these genes are responsible. And that particular time, there is no much multi-omics. Okay, only genomics was there. So I gave an idea. What I have mentioned, we have a genomic data. If suppose if we have a proteomics data, we can compare with the protein and genomics. And if we have a metabolites data, we can do the analysis. Then they have said yes, future definitely it will come, but now it is coming. If I have a metabolomics data of this particular plant, if I have proteomics data of particular plant, I have a genomic data. If I combine everything, I can validate, right, exactly these genes are responsible to produce this chemical. Yes or no? So that is what happening nowadays. The multi-omics gives the better results to predict each and everything, and we can do validation. So that's what I want to explain majorly on that particular topic. So there's a one more example of a proteomic study, which was my PhD topic.
Excuse
So
me, sir.
yes,
So I have a question regarding the previous slide. So
yeah.
totally, how much time did you take to complete this?
So actually speaking, when I was in college, 2014, we spent 3 months, 3 to 4 months. Okay, but in our industry, we completed in 1 day. Hope you understood.
Okay. And did you look for a particular secondary metabolite, or did
Yes.
you look at all the whatever the metabolites were produced? I
I'm
mean, the
going to
whole
show
cell.
that. In this project, we went with only one particular metabolites, and we did. But we can go with all the metabolites. What and all the metabolites process present.
Okay, thank you.
Okay, thank you. So yeah, I have my slides. I'll show that results. Okay, so the next image, this image represent one proteomic study of my PhD topic only on Parkinson disease I worked. So we have done quantitative proteomics studies of normal control and disease control of Parkinson's, and drug treated with low dose and drug treated with high dose. So then we have pulled all the proteins, and we have labeled. This is called TM-duplex labeling. After labeling, we have done the label check, and we have done the fractionation, LCMS, and data analysis. This is one of the mouse brain proteomics studies what I have done in my proteomics, my PhD topic. Okay, so this is one of the image of fast QC image. So how the quality is good. Actually speaking, the raw data is not up to the mark. The raw data is not good, but after trimming, the raw data has become good. So everything is in the green color region. Okay, but as of now, I cannot explain entire thing, but those are very basic things I'm explaining. And from our project, as I mentioned, there is a one oral cancer project, right? In the oral cancer project, we have selected one particular gene called CDKN2A. Okay, this particular gene or this particular protein is responsible for a disease is already proven. Okay, so we need to find out in this particular gene what and all the mutations are present, different type of mutations, and where the missense mutations are present. Okay, so that missense mutations were responsible for amino acid changes. That mutations we have induced, and we have done the molecular dynamic studies. If yeah, I have this data also, it's good. So in our project, we have taken CDKN2A gene. In that, leucine 16, the variant, leucine 16 changes to arginine. This informations we have taken from the literature survey. That means this particular mutation is already proven. This is more dangerous. In the CDKN2A, when 16th position, leucine changes to arginine, it is dangerous. It's already proven from the literature survey. But in our 12 samples, we found aspargine 71 changes to threonine and aspartic acid 74 changes to alanine. These two mutations are common in all the samples. It is not reported anywhere. these two mutations are more dangerous from the NGS analysis we found. Similar way, another gene we have taken, but I'm not going to explain this. These two mutations were identified from the 12 samples. Later, we performed molecular dynamic simulation. It comes under proteomics. CDKN2A wild type we have taken, and CDKN2A L16R. That means 16th position leucine modified to arginine. It's called mutated. We have done, and CDKN2A N71 changes to T, and CDKN2 D74 changes to A. These two mutations we identified from NGS. This mutation identified by already known articles. So then we perform molecular dynamic simulations using GROMAC software. We perform 300 nanosecond simulation. We found there is a magenta color, can able to see it's more fluctuations were there. It is not stable throughout the simulation. That represent the D74A, it's highly fluctuating. It is not stable throughout the simulation because of that mutation. That is what we concluded in this particular project. So similar way, we have done many analysis. I'm not going to explain much about this analysis. When it comes to the another project, I hope all of you know about that. There is
Sir,
one. Yeah. Yes.
so D74 mutation, you are showing that because owing to the mutation, the result was not stable. Is the mutation so frequently occurring?
No, it is in the. It is present in the 12 samples. In all 12 samples, that particular mutations are common.
Okay, so it's a record of all the 12 samples.
Yes, in a record of all 12 samples, but actually the 12 samples are very, very less. We need to go more number of samples. Then we need to conclude.
Okay,
You clear?
that's why this data was not in par with the other datas.
Yes, that's true. But we need to do a lot of things. And one more thing, that molecular dynamic simulations we perform for 300 nanosecond. Generally, what happened? People they'll go with 100 nanosecond itself. So in 100 nanosecond, how we can conclude? We cannot conclude anything in 100. Everything is almost high, low, and all these things. But we need to show at least 1000 nanosecond. Nowadays, this work we have done in 2000, I guess 18 or 19 before COVID, and that time we performed 300 nanosecond simulation. But nowadays, the research community they expect 1000 nanosecond or 2000 nanoseconds. You clear?
Yes.
Yeah, perfect. Thank you. So this project is I have shown one transcriptomic project, right? That particular data. So we have taken the plant called Androglophilus paniculata, and this data is approximately 11.2 GB data, which we downloaded in 2014. Okay, so try to understand. 2014 there is no geo downloading raw data itself. We need to rely on college internet. Downloading data itself is a big task. Okay, and the read length was 19 crores 75 lakhs 37,498 reads are present like this. Look at that. Greater than symbol first read, greater than symbol second read, third read, fourth read, like that. 19 crores reads were there. Each read consists of 101 base pair. So that means CTC, GG is there now, 101 base pair. Okay, this much data we downloaded, and we have done the different softwares. There are 3 softwares we perform. There is a software called Velvet, and CL's Genomic Workbench, and Trinity. Okay, just a minute, 1 second. Vidya, I'm in one meeting. I'll call you. I'm in one meeting. I'll call you later. I'm in one meeting. I'll call you. Hello. Yes. We have done with the 3 different softwares, and finally we found which just a minute, 1 second. Hello. Vidya, I'm in one guest talk. I'll call you later. Yes. So we found there are in that particular plant has 64,488 genes were identified. Because again I'm telling this, but when we started this work, there is no reference genome for that particular plant. Okay, the Androglophilus reference genome was not available, so that is the reason we have done de novo assembly From the de novo assembly, we found 64,488 genes and 86,215 transcripts were identified based on there's a tool called Trinity. Okay, then we have done gene ontology distribution for that particular data, and we found that particular genes are involved in what and all the function. We found most of the genes are present in the metabolic process. Can you able to see here metabolic process? After that, we have taken that particular metabolic data to confirm the pathway analysis. We have read the article. We came to know there is a one particular molecule called androglafloid, which is known anti-cancer and antiviral and anti-inflammatory activity molecule. Okay, now our idea is which genes are responsible to produce this particular androglafloid. So based on the literature, we came to know there are two pathways are there, MVA pathway and MEX-DVX pathway. So these pathways were analyzed using KEGG pathway. So it's opening now. Yeah, as you already know, we have glycolysis. Okay, in the glycolysis, right side we have DOX pathway and malmonite pathway. We have identified the transcripts, right, that we mapped against this database. So there is some green color highlights are there, right? These genes are responsible to produce diterpenide biosynthesis pathway that goes to diterpenide biosynthesis pathway to androglafloid. In the sense, the androglafloid is a diterpenide pathway. The diterpenide comes under terpenide pathways. The terpenide comes under DOX pathway. The DOX pathway starts from glycolysis. So we found what and all the genes are responsible for to producing this androglafloid compounds. That is what we concluded for this particular project. After that, to validate this our data is correct or not, we have done the phylogenetic analysis. So what we did, I hope all of you know about Rubisco. So there is a particular gene called Rubisco gene, which is common in all the plants. We have taken the Rubisco gene of different plants. Okay, please, different plants, and as well as Androglaphelus paniculata also. Okay, in the sense, Androglaphelus paniculata, Rubisco sequence were already available. Only that gene sequence that we have taken, and we taken our Trinity IDs. That means I told you now there are 86,000 Trinity's IDs were taken. All these sequence were taken, and we did multiple sequence alignment and phylogenetic analysis, and we came to know Androglaphelus paniculata similar to Trinity ID of this. This is confirming whatever we have taken the raw data, it is from Androglaphelus paniculata only. It is not from different sequence. To validate our results, we have done phylogenetic analysis. Okay, so yeah, till now, if you have any questions, please ask. Maybe because I have maybe I can say 10 minutes time. Yeah, if you have any questions, please ask.
So can you explain once more this phylogenetic tree?
So in the phylogenetic tree, what we have done to validate our results is correct or not. For that, what we have done, we have taken different plant Rubisco sequences. Are you clear? Only Rubisco sequence we have taken. Okay, and we have taken Androglaphelus paniculata Rubisco sequence also. It is already available in the database. Okay, but whole genome was not available, but only Rubisco sequence is available. And we have taken the R86,000 Trinity IDs. I told you now 86,000 Trinity IDs we found that we have taken, and we did multiple sequence alignment. Finally, we came to know Androglaphelus paniculata, it is similar to Trinity ID of this. That means whatever we have taken the raw data, that is Androglaphelus paniculata only, not with different organism. I hope you're clear, right? To validate, we have done this.
Hello.
Yeah, yes.
Sir, actually, Rubisco is an enzyme. It codes for the enzyme which is common in all the plants.
That's
And
what.
why did you particularly select the Rubisco for confirm to validate this your plant?
Because
On what basis?
Rubisco sequence is conserved in all the plants, right?
Yes, yes.
That is the reason
For
that particular plant should be present in our data also. In our data, it is present. And it
Okay.
is similar to androclassical paniculata only.
Mm-hmm.
To confirm that, we have done the phylogenetic analysis.
Okay.
Rubisco is an enzyme which is which is generated by RBCL gene. That gene sequence were identified in the Trinity ID. I hope you're clear, ma'am.
Yes, sir. Go ahead, please.
Yes. I guess this is my last slide. So if you have any questions,